Hello everyone, in this video we'll discuss about gene therapy and its overview. So, what is gene therapy? Gene therapy is a technique in which the normal genes are introduced into the cells for correcting the deficient gene or the abnormal gene, which are responsible for the genetic disorder or acquired disorder. Now, what are the types of gene therapy? One is the somatic cell line gene therapy. It involves the introduction of genes into the somatic cells of an affected individual. For example, the bone marrow cells, hepatic cells, and central nervous system. And the germ cell line gene therapy involves the transmissible permanent modification of the genome of a gamete, a zygote, or an embryo, in which the heritable disease can be cured for the upcoming next generations. But in practical life, the prospect of human germline gene therapy is not sanctioned yet. So here are some broader views about somatic cell line gene therapy. Somatic cell line gene therapy are being performed with some special vectors that have been engineered for gene transfer and are called the virus mediated transduction methods. So the viruses that are being used as a vector are like retrovirus, adeno-associated virus, lentivirus, adenovirus, and there are some major advantages so that the somatic cell line gene therapy can be performed easily by this virus vectors. Coming to the next slide, there is a brief idea about the retrovirus transduction method. Retrovirus, the first generation vector, contains three major proteins called the GAG, which codes for structural proteins of the virus, the pol or the polymerase codes for the reverse transcriptase, protease and integrase enzymes, and the envelope or the ENV codes for the retroviral coat proteins. The most commonly used retrovirus vector is based on MMLV or the mononeurone leukemia virus. As you can see the picture of the MMLV that infects and work on the rapidly dividing cells of the mouse and humans. The MMLV has LTR or long terminal repeat at each end, which controls the transcription and integration of the virus into the host genome. In step 2, it is necessary to make the retrovirus function as a vector. So the retroviral proteins, that is the three proteins GAG, POL and the ENV, are removed as it can put harmful influence and are replaced by the therapeutic genes. In order to produce vector particles, a packaging cell is essential. This packaging cell lines provide all the viral proteins required for the capsid production and the virion maturation of the vector. This packaging cell line have been made so that they contain all the three genes as shown in the picture. Coming to the next slide, that is the step 3, where the retrovirus means a virus that can have genetic material in the form of RNA molecule or in the form of DNA molecule while present in the host body. So when the virus vector enter into the host cell cytoplasm, it first transforms its genetic material into DNA from RNA by the process of reverse transcription and by the help of enzyme called reverse transcriptase. Another enzyme, RNase H or the ribonuclease H, represents a unique strategy to copy a single-stranded RNA genome into a double-stranded DNA. Here is the picture that shows the process of formation of DNA from an RNA molecule. Now, step 4. After the formation of DNA copy, the DNA copy enters into the nucleus and is free in the nucleus of the host cell. An enzyme called the integrase carried by virus helps in the insertion of the DNA copy into the larger DNA molecule of the host cell nucleus. But there are also some disadvantages of using the retrovirus vector. Like the integrase enzyme can put the genetic material of the virus in any arbitrary position in the genome of the host, which can lead to insertional mutagenesis or uncontrolled cell division, that is cancer. There are some other vectors for somatic cell line gene therapy adenovirus, adeno-associated virus. The adeno-associated virus is the most accepted vector in gene therapy in today's days because the advantages of adeno-associated virus are more than any other vectors used for somatic cell line gene therapy, as the adeno-associated virus integrates into the host cell's genome and provides stable expression. It has no immunity problem and it is non-pathogenic, and it infects both the dividing and non-dividing cells. Here is some brief idea about the diseases that are being treated by gene therapy. Number 1 is the severe combined immunodeficiency disorder caused due to the deficiency of adenosine deaminase enzyme, and was the first successful gene therapy. However, unsuccessful gene therapy can lead to leukemia. Number 2 is the sickle cell anemia that was treated by gene therapy. Others like hemophilia, blindness caused by retinitis pigmentosa, replacing the mutated genes, all this can be treated by gene therapy. I hope you liked this video. Thank you.